So what I'm gonna do, I'm gonna go through these questions and expand on it, what we can do to work out better instead of just going step by step. I think this makes more sense, and then we can kinda branch off. Okay, the first question, we have a picture of a membrane, obviously. Just one thing about this, a lot of times when students are answering questions or labeling things, don't put cell membrane, okay? They really want you to say plasma membrane, if you have a choice. Alright. What are 1, 2, and 3? So what is this supposed to be?
Glycoprotein.
Glycoprotein, okay. It's a combination of some carbohydrates and protein together, good. 2? Integral, and then where is 3? Oh, 3? Yeah, phospholipid head, right? So the correct answer was C. Good. You need to be able to draw and label this. So I'm going to go ahead and draw and label one really quick on the board because there's some things that are missing. Definitely draw some sort of integral protein when you get a chance. You can label it as both. So this is an integral protein that has, it's also a channel protein. Turn my volume down. That's distracting. Definitely the phospholipid head. Hydrophobic or hydrophilic?
Hydrophilic.
Right. And this is the, what is this?
Hydrophobic tail.
What kind of tail?
Yes.
Hydrophobic.
Yeah, it's hydrophobic, but
Or phospholipid tail.
it's the lipid, yeah? So this is the I should actually do this wrong. This is a phosphate head, I'm sorry. And this is the lipid tail. And together it's a phospholipid. So you also get points if you label the whole thing. What if I have a protein that's just on the outside like that? What kind of protein is that? Peripheral, right? It's on the outside. What if I draw some sort of structure in the middle like this that gives it stability?
Glycoprotein.
No, that's
Cholesterol.
that's one.
Cholesterol, good.
Wait, too much cholesterol makes the cell like too fluid,
It makes it less fluid.
or it's too stiff.
It holds it together. So a lot of times those cholesterol molecules are put in certain places to hold a group of proteins together that have to perform some sort of function so they don't float around the ocean. I put a protein pump in here. So if you want to do like a sodium potassium pump or something. Points for that. Am I getting anything? Okay, we did the glycoprotein good outside. Oh, coming off this. So that's one of the drawbacks that you need to know. All right. Next question. A single nerve cell does not think. Oh, we just talked about this. A single nerve cell does not think. Millions of them organized in the brain together. Help it thinking. What is this? Emerge, right? That's what we were talking about right here. So that's they have it almost every single year. They ask about this emergent property. You need all of them to work together. One single nerve cell can't do it on its own. What structures have phospholipid bilayers?
Nucleus and vesicle system.
So nucleoid is a region inside a bacteria that has the DNA. Definitely not surrounded by a membrane. Alright? So the correct answer, yeah, the correct answer is 1 and 2 only. What's a vesicle? So a lot of times I just draw it as a circle, but you really should think of it as a pinched off piece of the membrane. I'm gonna get lazy here, not do the whole thing. But in here you might have your protein, and this whole little bubble, this whole little vesicle might travel through the cell from endoplasmic reticulum to the Golgi, and then eventually to the membrane where it fuses and releases that molecule. What can be found in a plant cell but not animal cells?
Raw ER? Starch.
Let's look at these. Okay, so
What
of
is
course,
raw ER actually?
what's a rough ER?
Yeah, that's right.
So in a cell, you have the nucleus, and then usually right near the nucleus, you have this membrane structure that has ribosomes on them that will create proteins that will be used outside of the cell. So that's the rough ER. So little proteins are made in this direction, they put them in a vesicle, that vesicle travels to the Golgi, and then eventually to the membrane. Yeah, the correct answer is starch. Do you guys remember the two types of starch that you needed to know?
Cellulose?
Cellulose is not a starch. So if you remember, we're going to talk about this in biochemistry tomorrow.
Oh wait, starch is the one that makes like bread levitate?
No. Starch is in potato. Yeah.
This was a long time ago. Okay. So in biochemistry,
What's the correct?
in biochemistry last year, we talked about 3 different polysaccharides that you needed to know, 2 in plants and 1 in animals.
It was, in plants it was starch,
So one is starch,
cellulose.
and the other is cellulose, okay? So these are long chains of repeating units of glucose for all of them, or something similar to glucose. How about animals? Glycogen, okay, you guys totally remember this from what we were talking about, blood sugar control.
Yeah.
But they wanted you to go a step further down here. So there's 2 forms of starch that plants use, one for longer term energy storage, the other one for shorter one. One of them is shaped in this spiral helix shape.
Or is it alpha?
Alpha starch and then beta starch.
Nice, wouldn't it? And the other one is kind of branched, like glycogen, but not so complexly branched. So amylose was one, and the other one was amylopectin. But we'll practice this tomorrow. And then you had to know where the glucose molecules were connected. I don't know if you guys remember this. 1 to 4. You guys remember this a little bit? We'll talk about it tomorrow.
So Fleming, how can you remember everything?
Why did I teach you? Oh, Mr. Dorman.
Hey! Who's the best biology teacher in the history of EF?
These students
Hey,
are,
Gordon, good to see you.
they teach me. Welcome.
Want to grab lunch? You guys be around or you gotta take off?
Today, I could probably grab lunch, yeah.
What time? I'll come back. Give me a time.
12:15.
12:15, I'll see you here.
Okay.
Okay, sorry to interrupt. You guys are like, hey, Arsen, good to see you, man.
No. Alright. Next one, which pair of characteristics are correct for the cellular process of exo and endocytosis? Oh, did I skip one? No. We have a 100% agreement?
I thought it said.
So I think we're all in agreement that endo, we're bringing in, right? And exo, we send out,
Yes.
correct? Okay, so we got that. So exocytosis, this will get deep first. Exocytosis, vesicles will move towards the plasma membrane. Yeah, right, this is exo right here. So it's moving towards the plasma membrane. Will the membrane actually increase in size? It will, because what happens is, this vesicle actually becomes part of the membrane, so when it gets close, it will fuse to it, and then all of this will stretch out and it'll be the new membrane. So, if we were thinking about the processes that were happening here, this vesicle was at one time part of the rough ER, and then it fused with the Golgi apparatus, and then it became this molecule here, and then eventually spit out its protein. So yeah, it would actually increase in size. Do you get that?
But it's not DP though.
D's wrong?
Yeah, because the other side is endocytosis. and
Oh, it is, you're right.
So wait, which one is the?
So what's the right answer?
A or D?
Secretion of cellular materials, okay, vesicles are moved away from, yeah, right, they go this way. So it looks like A might be the best one. B, cell membranes fuse, that's correct.
I was right.
No, not B. Infolding, no, that would be endocytosis. Endocytosis is something, let's go this a step further. So there's something here that you want to bring in, and it touches the plasma membrane and it folds in and around it. So yeah, the correct answer was A. Thanks for catching that. Okay. All right, let's talk about diffusion. Let's first see how we did on this. So which statements are characteristics of diffusion through membranes?
2 and 3.
So polysaccharides don't diffuse, okay? You need some sort of larger process to get them in. Actually, polysaccharides you can't even bring in. So you have to bring them in as glucose, and a lot of times, like when we're making glycogen, they're condensed inside the cell, large molecules. So it can't be one. Facilitated by special channels, sure. Is affected by concentration gradients, yeah. So let's talk about the different types of transport we need to talk about, different types of diffusion. Called membrane transport. And then we broke it down into two main groups, active, and do you guys remember the opposite of active?
Like passive diffusion?
Passive. Okay, so those are the two main types of transport. So we have passive transport, which is no energy, and then active, which requires ATP. What else can you tell me about these two basic groups?
Passive is,
One is like channels,
No, what's the main thing though?
passive transport, like is towards the concentration gradient.
those were the.
Active is,
So we use these words like down the concentration gradient, which is high to low, and then active transport can pump it the other way. And here we say against concentration gradient. All right, so there were two types of passive transport that we needed to know.
facilitated.
Let's talk about facilitate. What facilitate means help. What's helping it?
Channel.
So here we have channel proteins that will help carry it across the membrane. Why would you need a channel protein? What's the deal with these molecules?
They are hydrophilic.
They are polar, right? They have some sort of issue, so they're hydrophilic. Can you give me example of a polar molecule that might need a channel protein? So definitely water. A lot of times students get tricked because like, oh, water goes right through. It doesn't, it's polar, it needs help. And we also, when we talk about nerve transmissions, sodium and potassium for sure. So those would be examples of molecules that need help with the channel protein. Okay, what about no help? Simple. They go right through the lipid bilayer.
So like CO2.
Yeah, examples are CO2, oxygen, those are ones that go right through. And then active transport, an example can be the sodium potassium pump. Alright, good? So the correct answer was 2 and 3, right? Okay, see. Be helped by special channels, it's definitely affected by concentration gradient. Okay, now we're into cell division, which usually takes the most time in the cell cycle.
Interesting.
So you need to remember this whole cell cycle little pie chart thing that we did one time in class. So if we were dividing this out, we can divide it into 3 main groups. There's one very large one. Then we have this shorter one, and then we have this even shorter one. So yeah, this larger one that takes up, I don't know, 80% of the cell's life, I don't know if that's accurate or not. This whole thing is called interphase. An interphase, we can divide interphase into 3 parts as well. What are the different phases of interphase?
Anaphase, telophase, metaphase.
So interphase, it's G1, S, and G2. You remember this? A little bit. What happens in G1?
The chromosomes are doubling.
That's here. So S stands for synthesis. So we're synthesizing DNA.
G1 to start dividing.
What starts dividing?
The DNA, like the nucleus.
No. We are making stuff in G1. We're making copies of things.
Copies of no, like the ribosomes and the mitochondria and organelles.
So we're getting ready for the big divide. We're getting ready for this big process over here called mitosis. So if you're gonna eventually create 2 cells, you're gonna need, it's like Noah's Ark. You're gonna need 2 of everything. You need more mitochondria, more ribosomes. So yeah, you're making more organelles. You're also doing things like increasing the size of the cytoplasm and all this other stuff. Synthesis, we're making DNA. What happens in G2? It's really not so exciting. So G2 just makes sure everything was done correctly. So it's like quality control. So we double check. So this is where you have these enzymes and they're going down the DNA to make sure it was copied correctly. I don't know if, I'm not sure if it's making sure the organelles were done right, but maybe. But definitely with DNA. Mitosis. What are the 4 phases of mitosis?
Anaphase, telophase.
Okay. What is the function of mitosis?
Like you said,
Yeah,
Is it?
to the body.
like cell reproduction. Oh no, growth and fix the injuries.
Identical what? So the actual real definition of mitosis is to create genetically identical nuclei. Because once you have two nuclei, you're still in one cell, but they say mitosis is finished. It's not till this last phase that you have your two cells. Alright, so if you see this, that is the best answer for mitosis. And this last little bit right here, cytokinesis, the physical splitting of the cell. Oh my god, we'll be in video showing how I can't spell. For the whole world to see. So when I first went live, the first person on it was Mohit. He was a third grade student of mine. Oh no, he was older. Fourth maybe. I might have
You used to teach in primary?
in Poland. So he's like, hey, Mr. Fleming's Mohit. What are you doing watching like a little chat? It was fun. Alright. So that's the cell cycle. So definitely the longest one is interphase, consisting of its normal. Oh, some questions that they love for this is like, which of the following happens in interphase? And like the answer is protein synthesis. So just remember, interphase, it's the normal life of the cell. It's doing things like transcription, translation, you know, protein synthesis.
Can you also talk about where it stops?
Where the stops are?
I mean, like, the checks, like, the things that, like, keep it between and this kind of stuff, that
Oh, you mean for like, okay, yeah, for
division.
controlling division?
Those,
Yeah, we're definitely talking about, that's like tumors and what controls that. Yeah, we definitely will. Oh, actually, while we here, do you guys remember the name of the proteins that control each one of these cycles?
Wait, they are wait, seconds.
Good, okay.
Wait, in G2?
Of all the cell cycles. All cell phases might be better. Okay, another plasma membrane. Which part of the plasma membrane is responsible for preventing the free movement of ions?
no,
So remember, if it's an ion, it's charged, right? It needs help to get across. So it needs this channel protein. And the thing that keeps ions from getting in here is that non-polar section of the plasma membrane.
So it's 4?
The correct answer is 4. The ion is like coming through and he's like, how do I get in here? I wish there was a channel protein. So there's no assessment statement that says that you need to know anything about standard deviation or t-tests. There is one for chi-square. But they want you to be able to understand this just based on the beautiful IAs that you made, okay, or the different labs that we did. So this one says, which population has the greatest variation in height?
C.
It is C. So remember that those are error bars that represent standard deviation. The correct answer is C. What is standard deviation?
the average difference of,
Like how far are the all values on average from the means?
I don't know if they're going to ask you this. This is the old syllabus, so I'm just going to put it on.
Standard deviation? Well, you know, I think it would be a math question.
So it's a spread of data around the mean. And there's actually a number. So one standard deviation, this math, did you guys do this? 66.6, whatever, 2/3 of the data is one standard deviation. So it's the measure of 66%, 67% of your data around me. That's one standard deviation. You go 2 standard deviations is 95%. I don't think I'll ask that. All right, facilitated diffusion. Passive movement, right, you don't need energy of a particle across a cell membrane with help with a channel protein, correct? Correct answer is B. What do you need for osmosis? Okay, we didn't talk about osmosis yet.
What is it, B? D. Oh, it's
D.
All right, so we have a partially permeable membrane, so that's the important part. You need something that some things can move across and some things can't. Usually in osmosis, we're talking that water can move across. So if I have these little circles I'm drawing, let's say that they're, it's sugar. Is that my solute or my solvent?
Solvent.
What was what?
I don't know, that's a solid.
That's a solute. So solute are the molecules dissolved in the solvent. And then here we have, water, which is our solvent. Which way is water going to move? To the left or to the right? To the left. So water moves to where the party is. Water moves to where the stuff is. So the definition of osmosis is the movement of water from a low solute to a high solute concentration. Can you think of an example where we saw this in human systems?
Kidneys.
Kidney for sure, right?
The lungs also, because like the higher concentration. No, but that's a kind. No, that's
That's
kind.
the movement of water, though.
But yeah, but still also like by this principle.
So yeah, the correct answer was D. What is naked DNA?
DNA is not supercoiled into chromosomes.
We got close.
No,
Is it C?
I think D.
Yeah, it's C.
Yeah, so the IB definition is actually C. So naked DNA means no proteins. No histones, no, I guess it'd be enzymes associated with it, but yeah. Where do we see naked DNA? Prokaryotes, yeah. That is a little confusing, right? Because prokaryotes also don't have a nuclear envelope. But the truth, the best answer is C. What do you call DNA that is associated with proteins?
Super?
Bacteria will be upset. What do you mean? I don't have DNA just because that's
No, wait,
nuclear?
Yes,
Protein. I've seen that one, right? Examples of some proteins would be like histones, for supercoiling and all that kind of stuff. Supercoiling for gene transcription. All right. Oh, this is like repetitive. Hitting you over the head. The function of proteins in passive transport? To serve as channels, yeah. All right. Here's some mitosis questions. Actually, you probably remember this from. So we have a micrograph. You need to be able to identify different stages of mitosis from an image. So does it ask what's the correct order?
Yeah.
Yeah.
So which one's probably the best for prophase?
2.
If it is, but it's maybe, does it say order or just prophase through telophase?
No, what order?
Okay, so yeah, so maybe that's not prophase, but early I don't know.
I think it's A or D.
I think it's D.
So So I think you're right, so two is the first stage. I'm gonna talk about some of the events that are happening in each one that you should know. We'll talk about these different stages. So what happens in prophase? What are some pieces of evidence that you would know by observing a cell that it might be in prophase?
Wait, there's no nucleus, right?
The nucleus begins to disappear.
DNA is like, spread over.
What do you mean? DNA what?
It's all over the side,
Like,
instead of being one.
it's not coiled.
Actually begins to, it's not quite like this where you can't even see it. I think it begins to coil. So you begin to see some evidence of actual chromosomes. That's how we see them in pictures. So coiling, supercoiling begins. Actually, they use the word condense. So those are the two big ones. The DNA begins to condense or form into those chromosomes that you can actually pull apart, and the nucleus disappears. How about metaphase? What happens in metaphase?
All of the line.
Right, so your chromosomes align on the equator, they say. How do they align on the equator? How do they move there?
They use
Telomeres?
two views.
So there's a lot of different terms for these. Sometimes they call them spindle fibers, also called microtubules.
Well, like, for example, in the flagella of the bacteria, inside there are like I just say, the microtubules that make them move.
In the flagella?
Yeah, I was,
Oh, but they're made of microtubules, maybe?
yeah.
Structure? Where do these spindle fibers come from?
Central.
So centrioles will move to the poles, so there's places for these spindle fibers to shoot from. Alright, how about anaphase? What are the main events in anaphase?
Pull to the folds of the
I think that's pretty much it. Chromosomes are pulled to the poles by those spindle fibers. And then telophase.
Once they split up,
The nucleus begins to reform. They don't, what splits?
the nucleus starts to split and then like they start to form new membranes.
That's it. So yeah, the correct answer you guys said was what? 2, 1, 3, 4?
Yeah.
Right? While I'm here, let's practice magnification. I'm going. What's that a cat? Oh. I'm going to measure this, and I want you to give me the magnification of this image, okay? Okay. So this is, oh, perfect, it's 10 centimeters. All right, go ahead. What's the magnification of this image?
That's micrometer, right? 25 micrometers.
That says 25 micrometers, yeah.
How many micrometers are in 1 millimeter? Like 1,000?
Yes. So I always like converting it to millimeters. I mean, just because I can remember 1,000 because my brain's so small. But yeah, 1,000 micrometers equals 1 millimeter.
Wait, 1 micrometer is equals to 1,000 milliliters, right? It's gonna be
Oh, wait.
40,000?
Oh, I'm sorry. No, that's right. Yeah. 1,000 micrometers equals
No,
1
no, no, I'm putting the magnification is 40,000.
40,000? I don't know. Check it out.
So, oh, so it's gonna be 100 divided by 250,000?
Yeah.
We should send it on testing that to Japan. We should try it. It's gonna be 4,000.
Times, I thought
So it's 10 centimeters, it's gonna be 100 millimeters, and it's gonna be. Is it like 100 by 250?
So check it out. So we said the ruler was 10 centimeters, but how many millimeters is that? 100, right?
It's gonna be 100,000 millimeters.
Divide by 25 times 1,000.
It's gonna be 4,000.
What's my units? They're really
Times.
X. They really aren't. I mean, you can put X there. But. That makes sense?
Yes.
Bonus magnification question. Free of charge.
Yeah, bonus check.
That's it. So,
There's a magnification question every year. Okay, so this is a little bit of 1.5, which is the origin of life. The origin of the first cell, I guess you'd say. So there's quite a bit in this regarding evolution. I know we did this a long time ago, but always good to review. Okay, the endosymbiotic theory says that eukaryotes arose from living communities formed by the merging of prokaryotic organisms and their host. Which of the following is the best evidence to support the endosymbiotic theory?
A.
Alright, so there was some sort of cell, so they think, that really didn't have life yet. They called them mycelials. And it was basically just an empty cell that was made of this phospholipid bilayer. And somehow, magic, bacteria ended up inside the cell. And if you look at your DNA today, we have DNA that's very similar to bacterial DNA. There's plasmids, there's,
so it's A?
correct answer is A. So, we have aerobic bacteria. They ended up becoming mitochondria. And then we have photosynthetic, I'll actually use the correct term, cyanobacteria are photosynthetic bacteria. They came in and they became chloroplasts.
I have a question.
Yeah.
The ribosomes in bacteria are SMDS, right?
They are. So are the ribosomes and mitochondria in your cells.
Also, like when we use antibiotics, we destroy the mitochondria.
Hopefully, I guess they're protected inside your cell. Yeah, because they would only attach to the outside.
The outside.
Yeah, how does it work?
I think there needs to be some sort of transportation for antibiotic to get inside the cell. I think we don't have that. But the bacteria that are already in your body, they're exposed to environment.
Right. Yeah. Makes sense.
That's what I think.
Great answer is A. What is another? Oh, yeah. And the last question here regarding the origin of life, which of the following gases is least likely to have existed?
Oxygen.
Oxygen, yeah. So there was no plants in the beginning of the world, so there were no organisms that could release oxygen as a waste product. So let's talk about some stuff that I may have missed on this quiz alone. Actually, you guys want like a 10-minute break and
Yeah,
sit here
actually,
since that-
yeah, like your phone has some kind of notifications.
Oh, does it? Yeah, take a little break and then, low battery, okay, close. Yeah, I need like, these it's too hard to see. Maybe.
When I was watching, I thought that the board was harder to see. Markers were pretty good.
Oh, you mean like, so if I turn the light off, maybe?
Yeah, maybe those ones. Yeah, but now markers are harder.
I need like multiple cameras.
And I think webinars work better.
So if I talked on a PowerPoint, like talk through a PowerPoint, is that what you're saying?
No, there's also this platform like YouTube that like they usually do conferences with, but you cannot communicate. It's not like Skype.
It's like chatting.
It's like this, yeah, but I think the quality is better because the traffic's lower on that platform. So many people on YouTube.
I need to, actually, it's not bad in full screen.
Yeah.
They are fine. No, two. But then it goes right to, I can record it. I guess it's being recorded, and I can just post a recording.
Yeah.
I don't know if I can edit it. I hope so. Let me know who's watching. Probably, I guess it's like my mom. My mother's probably like, how does he know?
This mesochasm thing is canceled? Not gonna do it anymore?
These are, I just can't get I don't know how they're supposed to do it. They want to, it's bizarre, I mean, it seems like I don't know what they want. So, it was like have students be able to assess, the sustainability of a mesocosm they created. So I guess you can just check oxygen and see how it.
Actually, I'm sure there's in Kickstarter, there's this new product where there's this thing, it's just a project now, they're funding it, called Transit.
So there's one that's working.
Yeah, but the thing is that, like, they give you the option to make it yourself. But you have to add the stuff to it. You have to feed your system.
Oh, so it's not completely.
No, it's not. But it works.
What's the black stuff?
I think it says, should be that.
And you're just using brine shrimp?
I think it's just the seeds, I mean the eggs.
So I tried to do that with my boy's fish tank. They have a fish tank at home, they've got a couple fish in there, but I'm trying to have like the shrimp be the decomposers and control the algae, and they're just not enough of them. So they just all day long, they're trying to eat all the algae, but they just can't keep up. They're like looking at me like, help. Can you please get some more? I gotta do a Kickstarter for the Bible, because I feel like so long, it's just changed.
It looks like a very new platform to start a business.
Interesting reviews, those stuff, they say it's more non-profit friendly. So what you couldn't use Kickstarter for non-profit?
They have like certain categories.
With education, the environment. Yeah, it seems to be more towards the arts, if I remember.
Arts and design, yeah. Global Nation Building Fund. This is non-profit. But it says like Global Nation wants to bring fresh universe instruments. But
Yeah, it's a movie.
this was all also really nice. They make this notebook, and then you have an app on your phone, and then once you mark under the paper, and then you use your phone to scan all these pages, and it automatically sends it to your Dropboxes and stuff. And then when you're done, when the notebooks fill, you can just microwave it, and it becomes clean again.
But seriously?
Everything you have to use a special pen, but it's like normal process. And we funded like pretty good, 2,223% after the thing they wanted.
So yeah, it's amazing how successful some of these ones are.
Yeah.
The other problem with Kickstarter, if you don't reach your goal, you don't get the money.
Yeah, it's for
I think Indiegogo, you get them like that. You, no matter what, you just get it, even though you're short. So you get kind of, no, you just get some. Why does this charger not want to work? Do you notice that? Like some chargers just don't?
I haven't had a problem.
No? I don't think I'm gonna win any Academy Awards for filmmaking. Personally, Yeah, so these are the juniors mesocosms, so I'm gonna have them, they took measurements on oxygen and CO2 and some
tape observations just on life in there, and then they're gonna open these things up, I'm sure they're gonna smell horrendous. And then make some conclusions about its sustainability, I guess. I'm not really sure what they want, though. Yeah, I just got a 6. Right, good.
You're late.
Are you?
Well, a little lunch is good.
You had lunch already?
Yeah.
A little bit.
No, I like it.
No, a little bit of lunch.
Free lunch,
Yeah, we're not gonna.
Pre-lunch.
an appetizer.
Yes.
Alright, just two things I wanna talk about, then I'm gonna have you do a post-test to see how much you've improved by. But, before we do that, I'd like you guys. Just draw a label. So I'm gonna get some new markers, because these are toast. Alright, so I'll give you prokaryote. Arshim, you have plant, and you guys can split the responsibilities for eukaryote. So I'm gonna draw a little one here.
Draw a little cell.
Yeah, draw a label eukaryotic cell. The IB way. Hey, Eduardo, you need a marker?
Yeah, you wait.
The IB way.
There's an ID for it.
Well, just about the labeling part, they want you to be neat and.
Eukaryotes are those ones who have nucleus.
Yeah. No, okay, give it back.
What is in the nucleus?
AES, right? ATS, right? Come on, dude. This work program. Properties. Let me see.
Can you get a point, just talk about the juicy liquid center?
What do we call it, vacuum,
Vacuole.
vacuum cell?
Yeah, it's energy storage.
Prokaryotes are bacteria.
Yes, and they have mitochondria also.
No, they don't.
What
They don't have anything that's an organelle.
Then that's it, I mean, we're done. DNA.
What do you guys do?
You can have them.
Right here, man.
You could also do, not drop, just
Wait, Mr. Fleming, when the trans is when hydrogens are on the different side?
We're going to talk about that tomorrow.
Yeah, trans is on the
Different.
opposite side, so. Alright, let's critique your art. Good, yeah, I think there's plenty of stuff like that. Be careful of the arrows though. I don't know if I would do arrows in order.
No?
I don't think I would do arrows. So I would just,
No arrows?
yeah, I've never seen them draw them with arrows. I don't think you can get points for this. Because what are you pointing to?
Hmm?
You pointing to the cytoplasm?
No,
How do I know? Okay, so make sure that you can definitely see that that's where it's going, okay? So I mean, that's a little bit big. Make sure that you're pointing to the dots, and maybe point to multiple ones, so that they know. Mitochondria is good, you have the way you draw them on the inside perfect. I think the lysosome's a little bit big. I didn't think I'm credit. Cytoplasm's good, plasma membrane, good, Golgi apparatus. I would even do a vesicle, if you wanted to do a vesicle going from one to the next. Actually, I've seen them don't award points if you don't show budding coming off the different structures. Showing that you understand that that's how the process works. But there's nothing else I can think of, I think it's okay. This is disaster. Good thing this isn't live. Oh, it is. So for prokaryote, I would draw, not all of them look this way, but I would definitely draw one with a flagella, just do it the normal way, the way I'd be liking to draw it. So there's a cell wall, but there's also the cell membrane. Actually, I'm supposed to have my flagella attached to the cell membrane. Like this, okay? So we label the flagella. What do you call all the little hairs on the outside that let it stick to things?
Pili.
Villi, right?
Yeah.
What do we call that region inside the middle of it? Nucleoid region. How many chromosomes make up the nucleoid region of the DNA?
How many chromosomes? That's what it's.
One. There's only one chromosome, it's one really big circular piece. What do we call the circular DNA?
Plasmid.
Those are plasmids, good.